Well, pleasure to be with you all here today. And it's a pleasure to present our exosome technology that's been developed at Rion. I started doing stem cell research about 25 years ago as a graduate student, and really the pluripotent stem cell, or the discovery of embryonic stem cells in the late 90s, captured a lot of the imagination of graduate students and medical students alike. These cells really hold the promise of forming any tissues, but they're extremely difficult to actually put into the human body. So as an alternative, the field rapidly evolved from trying to build the building blocks of tissue regeneration to using cells that could potentially modulate inflammation and promote healing from the body itself. Technologies such as PRP and mesenchymal stem cells started to evolve. And over the last 20 years, me and several other colleagues at the Mayo Clinic have been leading clinical efforts to deploy these stem cell-based technologies, or PRP, in an effort to resolve diseases that we felt are difficult to treat. However, despite this long-term effort through use of MSCs, we have not yet been able to achieve FDA approval. Right? The cell-to-cell inconsistency, the lot-to-lot variability, the inability to scale these technologies, and the massive cost of cell-based technologies just don't let us move this forward, and there hasn't yet been an FDA approval. So out of frustration, we started looking at Mayo at what is it about stem cells that captures our imagination? Why does it work sometimes? And why is it that they fail a lot of times? And when we did our large heart failure study called the Chart-1 study, around 400 patients, biggest heart failure trial to date with stem cells, we discovered that, as we've seen before, 5 to 10% of patients have this remarkable result. I'd bring them up on stage here with me. You'd all clap that they've done so well. But the rest of the 90% of patients don't respond. And so we biobanked the cells from each of those patients in the trial and found that indeed that needle in a haystack does achieve high degrees of regeneration, but that most don't. And the ones that achieve regeneration were secreting a particular exosome phenotype. And we were very excited because we thought, oh, okay, look, the MSCs are making these exosomes. We're going to purify them. Turns out that these exosomes weren't being made by the MSCs. They're being recycled by MSCs, and they're made by platelets. The MSCs just grab them from the serum and the culture medium and recycle them. That exosome innovation and discovery allowed us to now scale this technology such that we can work towards treating every patient with a uniform regenerative therapeutic. And that's really the genesis of Rion. So when we discovered all of this at Mayo, we were able to patent these proprietary processes and then license them exclusively to Rion, and pivoted the effort into the corporate architecture. So if we think about the field now, we went from a high-cost, expensive, autologous approach that's variable and non-scalable to now really a next-generation platform that's low-cost, massively scalable, and can be used in any office on the planet. And so when we think about extracellular vesicles coming out of platelets, we have this biomanufacturing process that renders these exosomes, these regenerative exosomes at scale, that allow us to pursue all aspects of soft tissue healing in the body. Obviously, I'm very interested in cardiovascular regeneration, but we know that this product is truly made in the body for skin healing. And so we got to go where the science leads us. So why platelets, right? If you think about it, when you injure yourself, what's the first responder to that injury? It's the platelets, right? They come when you bleed. The MSCs are already in your tissue resident. They're waiting to hear the message from bleeding. So the message comes from the platelets. It activates your stem cells that are already residing in your tissue, and regeneration ensues. The regenerative exosomes from platelets, or what we call regenosomes, do four things. They are massively antioxidant, they reduce local inflammation, they induce angiogenesis, and they offer a cue to local stem cells to divide and help repair tissues. The other very important thing about exosomes, and this is one of the reasons why they've been hard to purify and to store on the shelf, is that conventional processes actually render them fractured. And so you lose the contents of these exosomes. You break their shell, and as you break the shell, you have to flash freeze them and keep them frozen. Our process actually preserves the shell of these exosomes so that the contents never release. So we can lyophilize this product and leave it on the shelf for 2 years at room temperature without any loss of function. So taken together, we actually have scaled this technology now such that within Plated Skin Science, which is the skincare product, we put 1 trillion exosomes per ml. We know that you need at least 250 billion exosomes per ml to get any biological activity, even in a petri dish. And the 4X product, the plated product, has 15 cc's, so it's room temperature stable, so you can give it to the patient and they can use it prospectively. We leveraged this technology in a trial at Mayo called Platelets on Skin Health. 56 subjects were treated. Saranya Wiles, who's in the audience, was the PI for this study. The 6-week data was presented in Maui Derm, and I'll give you some sneak previews of the 12-week data. Most of it's embargoed because of peer review. But what we see is dramatic reduction in redness. We see dramatic improvement in even tone, and overall, we see really significant improvement in skin health. And when you start looking under the hood at the ultrastructure and histology of the skin, you see improvement in collagen content, with the exosomes actually stuck, if you look carefully, on the collagen fibrils in the dermis. We see improved collagen thickness significantly. And when you look at the histology of these patients, you can see more blue. Blue is collagen, so lots more blue staining, and the black staining is elastin, so significantly improved elastin content at 12 weeks versus baseline. Beyond the skincare space, these exosomes in the aesthetic space are being now looked at for scalp health and hair transplantation. We have the opportunity to leverage these with hydrogels like the ones you just heard about and the ones you will hear about as a biopotentiator and really contributing to the filler space. So overall, stem cells are beneficial, but it's rare to see them be of benefit. The platelets are the key source of regenerative exosomes, and we've been able to purify those and really scale them as a room temperature stable, off-the-shelf technology, which now in the POSH trial we showed has significant benefit on skin health. Thanks very much.